Welcome everyone to the second webinar of the Joint Dissemination Network webinar series. We had our first webinar on the 5th of September. The recording is now available online for those who missed it. But of course, you're here now for the second one, and we'll also have a third one. I have some more information about that later. Sorry, there we go. I have some house rules for this webinar. You can ask your questions in the chat box, or because we today have all presentations from the same project, I think it would be nice to do the Q&A at the end of the session. So you can either post them in the chat box or you can ask them in person at the end of the webinar. If you have any technical issues, feel free to post a message in the chat box as well, or send an email to my colleague, Maureen McDonald at fap.info. While people are presenting, it would be great if you mute yourself so we don't have any odd sounds happening in the meanwhile. And please note that we are recording this session and we'll be sharing it on our website and social media. So if you don't like to be recorded, it's better to leave the meeting and wait for the recording to become available. Those who were here last time have already seen a little bit of this, but I'm going to do it again anyways. My name is Noralie. I am a project manager at European Forum of Farm Animal Breeders. This organization serves as the voice of animal breeders in Europe. We ensure the representation of our members at European level. We are involved in the European policy and legislation as part of farm animal breeding, but also reproduction. We support and promote responsible and balanced breeding with our own code EFABAR. And we aim to promote dialogue around sustainable animal breeding and farming. Of course, this webinar is part of that, but most of this webinar is part of our task as a knowledge provider in European projects. On the slides here, you can actually see some of the projects that we're currently involved in, and some new ones are coming. HoloRuminant, the organizer of this webinar series, is one of them. And next to this network of projects and supporting our members, we are also running the Farm Animal Breeding and Reproduction Technology Platform, which helps to shape the research and innovation agenda and to set the priorities for animal breeding and reproduction in Europe. We do this by connecting knowledge institutes and the private sector. And as already mentioned, we promote and support the research and innovation in animal breeding. That's a little bit about me and the company I work for. Then a little bit about the HoloRuminant Joint Dissemination Network. We've set up this Joint Dissemination Network with an aim to provide an environment to optimize and share efforts and communication materials and resources. So this webinar series is one of the first things we're actually doing with the project group. But there's hopefully there's much more to come, because we have also applied for a Horizon Results Booster. So we'll be experts in disseminating all things related to the Joint Dissemination Network. In this network, we currently have the following projects. I'm not going to explain every single one of them, because during all the webinars, you'll hear a little bit about what they do. But these 5 projects represent a complementary effort to reduce the environmental and climate footprint of livestock production. But there are also different focus points, such as genetic diversity, efficient production, and, well, I think the mitigation of greenhouse gas emissions is basically the big threat between the different projects. In the first webinar, I've already heard from HoloRuminant, Rumigen, and Livestock. And today we'll basically have a 3D omic special, so I hope you're ready for it. Here's an overview of the presenters we're going to see today. And we'll start with Antton Alberdi with his presentation, diving into microscale host-microbiota interactions. So with that, I think it's time that we give the floor to Antton. There we go. I'll stop sharing.
Thank you so much, Noralie. Can you hear me?
Yeah.
Excellent. So I am Antton Alberdi. I am an associate professor at the University of Copenhagen, and I am coordinating this European project, part of this dissemination network, which is 3D omics. I will try to share my slides. Let's see if we can make it work. Can you see the slide? I cannot see you, so if you can say something, that would be helpful.
We see the presentation.
Excellent. Thank you so much. So I will give you an overview of what is the background, where 3D omics is coming from, and what we are doing 3D omics. It's going to be a very shallow introduction, setting the framework, and then Jenny, Victoria, and Bryan will delve into the actual action that we are conducting in this European project, which, by the way, it's already 2 years in. So we are in the 24-month landmark this month, actually. So I hope I don't need to convince you about the importance of microorganisms associated with animals and plants and any other eukaryotic organism in the many biological processes that they need to conduct for their development, for interacting with the environment, regarding the nutrition, regarding reproduction, etc., etc. And this is the main framework which we are when talking about 3D omics.
Sorry, Antton, I think you're seeing your presenter mode. I think you need to switch screens.
Okay, let's see if I can do that. Let's hope that perhaps if I share the entire screen might be the easiest way to do that. Wait a second. If I share the desktop, let me know if now it looks okay.
Yeah, now we see
yes.
just the screen. Yeah.
Yeah, okay, but you see the slide, right?
Yes.
Super. So what's one commonality of all these host microbiota interactions are that they're complex interactions, very complex interactions. And one way we have tried to go into this understanding these interactions has been to improve the resolution of the molecular information that we have generated, but not only characterizing the prokaryotic genomes or the expression, and also characterizing the proteins that they encode for, the metabolites that they produce as a result of enzymatic reactions, and do the same also at the host level. And this is something that many researchers nowadays are doing or trying to do, including ourselves. But when we started thinking about 3D omics, we thought this was not enough. Even though it is complex, one of the main limitations that we have when we do this is that although we can get a lot of very high resolution information, like, let's see if I can, now for some reason I cannot pass the slide. Okay, although we can get a lot of information about the functional capabilities, the functional activity of the bacteria that are associated with these animals. And we can do very cool modeling stuff to understand what is the actual contribution these bacteria are doing to the animals. We always miss one key information, which is the three-dimensional arrangement of bacteria in the intestinal tract. We go into very detailed informations, but without considering at all the spatial resolution. And this is something we are trying to address in 3D omics, because we know that microorganisms do not arrange randomly within the gastrointestinal tract. There are some bacteria that feed on the mucosa, the mucus that the intestinal wall produces. There are some other bacteria that feed on feed particles. There are some bacteria that rely on other bacteria for interacting and for getting the nutrients that they need, etc., etc. And in 3D omics, what we are trying to do is to analyze all these interactions at the scale they are happening, rather than getting one single sample for the entire intestine or getting a few samples for characterizing the intestine. Take thousands of samples per intestinal sample that we collect. And that way, try to reconstruct a three-dimensional landscape of the microbial microbe interactions and microbe host interactions. This is the main structure of the workflow So we are running experimentation trials with animals, both with chickens and with pigs. And once we get these samples, we embed intestinal segments into some embedding material that's then to keep the animals or the samples frozen. We cryoslice these frozen materials, and after getting very thin layers of the intestine, we use laser microdissection to collect very tiny sample sizes, so very tiny samples from these intestines. And then we do kind of single cell omic-like analysis to characterize what is the composition of the microbial community at this level of spatial resolution, and also what is the activity these microorganisms are conducting at this scale. Although we have not reached yet the later stage of the project, the idea then is that once all these data are generated, to do modeling so that these spatial interactions can be resolved. The project comprises 13 participants, 6 universities, 2 research centers, and 5 companies, all based in Europe, and 1 participant in Israel. And this is the main structure of the project. We have 2 work packages that go throughout the entire duration of the project, and then we have 2 or 3 main sections. The first one is about the technology development, and this is the phase that we are concluding now, where we've been developing a lot of tools for generating high quality information, for analyzing this information, etc., etc. We have started with the second section, which is the technology showcasing. We are actually right now running, we have just finished the second trial, and we are about to start, well, we have already started the third trial, the big scale trial that will provide us with the samples that we will then analyze using both macro scale conventional multi-omic methods and also the micro scale methods I was talking about. And once all this data are generated and analyzed, our plan is to assess what is the impact of implementing this kind of new technology in the analysis of microbe and host microbiota interactions, and the new knowledge we can generate for improving animal production, for improving breeding, for improving sustainability of production, etc., etc. This is one of the most important infrastructure pieces we have here in Copenhagen, and this is what we are using for generating these microscale samples, which is an LMD7 microscope from Leica. And this is just to show you that's how the samples are generated. Bryan will give us an overview of this in the third talk, I believe. But before Bryan takes us to the microscale, Victoria and Jenny will give us an overview of what we are doing at the macro scale as well, because we have generated many, we have carried out many experimental trials. 3 of them have been conducted in poultry, 2 have been conducted in swine, and we will learn more about this in the next talk. I will just give you an overview of what is the interconnectedness of the project participants, because it is quite complex. It is quite unusual in European projects to have this large degree of interconnectedness and codependencies, which means that we very much depend on what others do, and that requires to have a very smooth communication and management strategies, because the partners that are running the experiments need to provide other partners with the samples that will then generate the data that other partners will analyze. And so far, this has been, even though of course it has challenges, it has been running quite smoothly. And we hope that in the end, all together, we will be able to generate some new kind of data that will really make a difference on how we can understand microbe and host microbiota interactions. Just to finalize this initial overview, this is a picture we took last March in Barcelona when we gathered together for our 3rd General Assembly, I believe. We are having our next General Assembly in Oslo the next week. So this wonderful team of people is essential for this project to be being carried out as well as it's been happening so far. So I would like to thank them all for their contribution. And thank you all as well for your attention. So I will now stop sharing the screen if I can do that. and pass the baton to Jenny and Claudia. Or Jenny and Victoria, sorry.
Thank you so much, Antton, for the introduction to 3D omics. Yeah, as said, we'll do the questions at the end. So indeed, let's move to Jenny and Victoria. The floor is yours, guys.
Yes, let me know. Are you able to see the screen in?
Yeah, it's working.
Right. Great.
Thank you very much. So I will give it a start, as Antton already introduced and gave a broad overview on the 3D omics project. Jenny and I will now give a bit further focus on the in vivo models that we are working with. And myself, I will start with the poultry models. My name is Victoria Drauch. I'm a veterinarian and postdoctoral researcher at the clinical unit of poultry medicine in the Veterinary Medicine University in Vienna. And our task was starting actually, yes, you can switch, Jenny, please. Was starting as Antton already said, the technology development. And this was done by defining optimal sampling procedures and also assessing the effect sizes. So we had a proof of principle poultry trial, which was done in a small scale. The pathogen that we used was Salmonella infantis, and we included a multi-species symbiotic product called Poultry Star. And on the right side, you can actually see the setup. So each group consisted of 15 birds. We had two negative groups, T1 and T2. One of them was given a symbiotic product, the other was not. And we had two groups which were infected, same setup, one of them with the symbiotic product, the other one without. And the symbiotic product was not only given into the feed, but also into the water. And birds were infected on day 7 of age. And then we had weekly sampling points to actually collect cloacal swabs as well as the intestine. In regards to the intestine, which you can see in the next slide, is the sketch of a chicken intestine itself. And together with this sketch, you can actually see the samples that we were gathering. And with the color code, you can see what those samples were used for. So for example, in green would be the samples that were used for fish at UCPH in Copenhagen, which will be explained by Bryan further. In yellow, you'd be to see the samples that we took for colony forming unit count. So as we're working with the bacteria, this is how we measure the density. In red, you can see samples of digester that were taken and shipped to ETH in Zurich for in vitro analysis. In blue would be digestive mucosa and tissue of one sample that was sent to UCPH for multi-omic analysis. And in purple, we took digester and tissue and sent it to Affecta for metabolomic analysis. And that leads me to the main trials, so the big scale trials and the poultry health challenges that we are analyzing and focusing on. So the first one, including the bacterial pathogen Salmonella infantis. And here we are also focusing on the already named symbiotic product, Poultry Star. We had 3 groups where we added the Poultry Star, 1 into water and feed, 1 only water application, and 1 group with only feed application. Then we had a group that was only infected but not given the symbiotic product, and the last group as a negative control with no infection and no symbiotic. So this trial actually finished recently. And the second poultry health challenge that we're focusing on is on the viral pathogen, a fowl adenovirus. And here we include the impact of antimicrobial treatments. And this will be done in 2 different chicken breeds, so layer chickens and broiler chickens. 2 groups will be infected and given antimicrobials. 2 groups will be infected but not given antimicrobials. And then we again have the 2 negative control groups. And the third poultry health challenge is with the parasite protozoan pathogen named Histomonas meleagritis. And here we are focusing on the impact of the pathogen and a prototype vaccine. And this will be done in turkeys and chickens. So again, we have two groups being vaccinated and infected, one group only infected, and one group not infected and not given the vaccine. Yeah, and this is the setup of the 3 poultry health challenges. And with this, I would like to give the floor to Jenny.
Thank you so much, Vicky. So yeah, my name is Jenny, and I'm a PhD candidate at NMBU in Norway. My tasks within 3D omics are mainly bioinformatical analysis of the omics data that we get from these pig models. And I'm also helping with coordinating the final pig trial, which is currently running here in Ås. And today I will attempt to give you an overview of the pig models that we use in 3D omics. There are a total of 4 pig trials in 3D omics, and they are divided into 3 categories. The first one was completed in Copenhagen last year as a proof of principle trial. Here we focused on refining the experimental conditions and methods that would be used later on. We also collected the initial biological samples used for developing the data generation techniques. And additionally, since this trial used the same experimental factors as one of the large-scale trial, this small experiment also provided data for initial investigations of the effect that this prebiotic fiber called mannan may have on the hosts and their gut microbiome. Secondly, we have the main mannan trial, which is conducted so that we can see whether the fibers could help modulate the microbial composition of the piglet gut, as this can make the animals better adapted to the solid diet that they receive after being weaned from their mothers. Lastly, we have 2 replicates of a trial, which also investigate the effect of diet on the microbiome, but now focusing on the interplay between the host's protein deposition efficiency, the protein content in the diets, nutrient digestibility, and the microbiome. We aim to gain some insights into how we can use these factors to improve the nutrient uptake from the feed while keeping the animals happy and healthy. In the next few slides, I will give you a bit more detail into each of these trials, and then I will go into a brief overview of what the samples we collect from these different trials are used for. So the proof of principle trial was conducted last year in Copenhagen. It had 15 piglets divided into 4 groups, which were used to test 3 experimental factors. All of the groups were given the same basal diet, and then 3 groups were given the mannan fibers as a prebiotic additive to this diet. This is the main contrast of interest, control versus mannan, and we will recognize this also in the later trial in the next slide. However, we also included some additional factors that could have been implemented in the later trials. For one, we tested whether the piglets could benefit even more from the mannan fibers by having them added to their diet at an earlier developmental stage. One group therefore received these mannan fibers a few weeks before the piglets were weaned, with then the fibers being dissolved in their milk. After they were weaned, all of the 3 mannan groups then received the fiber added to their solid feed. Secondly, one of the groups that only received mannan after the piglets were weaned received a different fiber variant than the others. This fiber variant had been modified by the addition of a fluorescent label, which makes the fibers much easier to detect in downstream processes, and it's therefore the preferred variant to work with, although we don't know if it may bias the uptake and digestion of the fibers. But with this contrast, we can now investigate whether this label biases anything. So moving on to the main scale trial for the mannan experiments. In this 5-week trial that was conducted here in Ås earlier this year, we had 24 piglets that were simply divided into 2 groups. All of them were given the same basal diet, and then one group was given the Manon fibers that we saw previously. These are the unlabeled Manon variants. And then we can see whether these different diets introduce any differences in the pig health, the microbiota composition, the microbial activity, and so on. Lastly, we have the trials that investigate the interplay between host genomics, dietary protein contents, and the gut microbiome. We have 2 replicates of this 13-week trial occur here in Ås. The first round lasted from May till August this year, and the second one started in August and is set to complete in November. For each of these replicates, we have 2 groups of 30 pigs each, which have 2 different potentials for their protein deposition efficiency, which is determined by their genotype. We then combine this genotypic categorization with 3 different diets with sets protein content, either low, intermediate, or high. Then we can investigate how well each pig genotype makes use of the available dietary protein, how well each different feed is digested, and how the gut microbiome may differ across genotypes and diets. For my final slide, I'll summarize what the samples we collect from these trials are and what they will be used for. In all of the large-scale trials, we collect fecal samples from all of the animals before, twice during, and at the very end of the trial. These samples give some insight into the feed digestibility for the protein trials, and the nitrogen content, the microbial composition, and the microbial activity across all of the trials. And we can use these data to study the changes in the gut microbiome as the various experimental contrasts and as a time series. The endpoint samples are collected from selected euthanized pigs, and they vary from collecting digestine mucus from inside of the intestine to extracting specific sections of the gastrointestinal tract. These tissue samples are used for developing the 3D-omic technology and for complementary analysis like fish, which Bryan will talk about later. We also collect microbial samples here, and these are used for in vitro experiments. And with metagenomic and metatranscriptomic analyses, we look at the microbial composition and activity using what we refer to as conventional omics methods, which is MIMA and task. Then incorporating the additional layers of host genomics, transcriptomics, and metabolomics, we hope to investigate how the pigs and their microbiome as holobionts in a more complete picture may respond to the various experimental contrasts that we have subjected the in vivo models to. Yeah, so that was a relatively brief overview of the in vivo models. I hope you've got something out of it. Thank you so much for your attention.
There you go. Thank you so much for this presentation. I think we're just gonna move to Bryan and then go.
So, hi everyone. My name is Bryan Wang, and I'm a postdoctoral research fellow at University of Copenhagen. And so I'm basically going to walk you guys through what the project has been working on for the past 2 years, kind of building up on what has been introduced already. And so earlier, Antton had showed you this general workflow of the whole experiment, and Jenny and Victoria have both shown how their roles in the animal trials happen upstream of kind of what I'm working on. And so the part that I'm really focused on is highlighted in yellow, which is the microscale level of studying all of these samples. And so basically, after Victoria and Jenny do their animal trials and everything, analysis and whatever, they sent us different tissue samples, frozen, flash frozen, from their place over to us. And then I'm in charge of basically taking these tissue samples, cryo-sectioning them with a cryotome at a very low temperature, and then using a laser microdissection microscope to cut very, very small samples. And so Antton had showed you earlier, this is what a typical laser microdissection microscope looks like, But the benefit of 3D omics is, in collaboration with Leica and Ocolabs, which is a company in Italy, they've made a state-of-the-art, one-of-a-kind microscope setup, such that this laser microdissection microscope is not only in a cold room, but it's also in a cooling chamber that's able to cool the whole setup down to 0 to 4 degrees. And so because we're very interested in metabolomics and also RNA, which are famously things that are volatile, the cooler we can do our workflow, the better we'll be able to retain all of this information in our analysis. And so this, again, is the workflow that I'm working through. We've now mentioned fish a couple times, which will come down in imaging, but also there's the microdissection part. So as Victoria had shown, this is how a chicken fat kind of looks like. And so they've sectioned out all these different specific parts, both for traditional multi-omics, but also for what we're doing here. Basically, once we have a tissue section, the first step is to cut it into a smaller section. We embed it into a plant-based cryoprotectant. In our case, it's CMC, which is carboxymethylcellulose. Then we flash freeze this, and now we're able to cryotome it perpendicular to where the track face is. And so basically, we keep track of whether or not we're sectioning from proximal to distal, so we can figure out where each slice of cryosection is coming from. And so these subsequent cryosections, this is what it looks like, a very beautiful cryosection of a healthy chicken cecum. And so with our microscope, you can also zoom in and start to see that you can very quickly tell where there's host tissue, but where there's also microbiota. And so with this microscope, we're able to cut out different parts of interest and study what is there. And so the idea is to have these sequential cryosections go to different analysis pipelines so that we can figure out what is the DNA that is in this cryosection, what's RNA, or what are the metabolites. So here is on the bottom right, an image of these 3 cryosections on a frame slide. And the Leica software is really cool. I often tell my students that it kind of feels like a computer game because you're basically putting on these cryo samples and you're shooting a laser that cuts out where you tell it to do. And so if that's not a computer game in science, I don't know what else is. And this is how it looks like with the software. And because we have different objectives, different sizes, we're able to cut as small as 10 micron, which is about, if you do the math, is about 10 bacterial cells. And the idea is to, with these microsamples, we analyze their different metabolomes and whether or not different bacteria are next to each other, etc. And this is kind of what it looks like. In the microscope, it almost looks like you're copy and pasting a thing and cutting it out. And so on the right, you see that this is a villi that has been stained, and we basically just took it out entirely and analyzed it. So after we've done all this cutting, we've mentioned that this is a very large collaboration with people all over Europe. And what happens is we cut these out, freeze these samples, and then we send them to experts around Europe. And so there's a lot, as we are going on, as the project's going on, we're still pushing each of our technology to see how low, basically what is the limit of detection that we're able to see resolution of DNA, RNA, and metabolites, and that is still ongoing. And basically, the lower that we can go, the better the resolution. Basically, your eyes will be better to see at this specific region what is happening, and so on. But so while we were optimizing all of the tools, we also had a very preliminary kind of experiment where we used these microdissections and we just did a very standard 16S analysis. And this was in working with my student, Carlos, who's now back in Barcelona. And so the idea was basically to benchmark with already available tools, what can we see? And basically, we took these microdissections, purified them with a very standard DNA isolation kit, and then ran a 16S analysis. And we know that this is just a benchmark, and that the experts and the tools that we develop downstream will give us more resolution and all these. But this was just to get an idea on whether or not we're able to just get DNA from microdissections. And so in order to do this pilot study, Carlos and I looked at 2 cohorts of chicken. They unfortunately came from 2 different trials, so we can't compare them laterally too much, but we can compare them within the trial. And so we had selected 4 healthy chickens. We took their ceca and their ileum, which are 2 different parts of the gut. And then we took a cohort of 4 salmonella-infected chicken, also took their cecum and ileum. And from that, we were able to PCR off the 16S amplicon and look at community divergences. And so the first thing we looked at was whether or not the diversity of microdissections that came from these areas were different. And so immediately you see that the ceca of both the healthy and the salmonella-infected chicken are higher in diversity, alpha diversity. And this has previously been published already, that we expected there to be more cecal diversity than in the ileum. But what's interesting is also that the cecum between the healthy and the salmonella-infected chickens seem to be also different, because it has been shown that microbiome diversity is also an indicator for infection onset. And then another interesting thing is, if we look at the beta diversity of these analysis, we do see that the clustering of healthy chicken versus salmonella chicken is pretty different, which means that the microbiota are made up of different bacteria. But also, if you zoom in a little bit more, when you compare the microbiota of healthy chicken, healthy ceca versus healthy ileum, they're pretty different. But when you look at this context in the salmonella chicken, the salmonella-infected ileum seems to be a subset of the ceca. And so from this data, we can say that it seems like salmonella infection shifts the diversity of the ceca closer to its ileum, which is not the case in the healthy chicken. And then also with these type of analysis, we're able to look at abundance plots. I'm not gonna walk you all through this too much because there's a lot of detail, but if we look at just differential abundance of a healthy chicken versus that of a salmonella chicken, there are different clades that seem to pop up immediately. And so I want to draw your attention specifically to lactobacillus, because in this analysis, very preliminary analysis, we show that we can only see in DNA-wise, we can only see lactobacillus in the healthy chicken, but never in the salmonella chicken. And so hold on to that piece of information because it'll be relevant. And so this was just a very preliminary workflow to see whether or not our laser microdissection, cryosectioning, and all of these processes can give us viable DNA for analysis. And this proves that it is able to. But the second part of this presentation is basically talking about fish analysis, as my colleagues have explained. And so fish briefly is fluorescence in situ hybridization. And it is the concept of using a fluorescent probe to fish out, okay, sorry for the pun, to figure out the different targets that you're looking for and look at it with fluorescence microscopy. And so here on the right, you see a panel of a patient biopsy that has been fished for bacteria in general. And you see that these signals actually indicate that there's a bacteria infection in this biopsy. And so with such a tool, we're able to ask questions about what bacteria are next to each other, and what they're expressing eventually. And so a lot of the work that I've been doing in the project is optimizing different complex fish protocols to work on our cryosections. And so you can imagine a lot of times in microscopy, you're limited by the number of fluorophores you have. But now with advancement of imaging technology, image analysis, etc., we're able to do combinatorial labeling. So instead of just using one color for one thing, if you mix different colors and make colorful barcodes, you can increase the number of IDs. And so I'm pretty excited to report that so far we are able to complex 4 fluorophores up to 15 IDs. And I'm also pretty excited to report that the fish protocol that we have seems to work pretty well. not pretty well, works really well with our cryosections. And so this is an image of a healthy chicken cecum cryosection where we've now fished for all the bacteria. And when I saw this image, I was particularly very excited because if you zoom into this image, not only do you see thousands of bacteria, which is what I'm interested in, but you also see there are these pockets of localized brightness. And so these two aggregates of cells, I'm willing to bet for now that they are clonal populations of a single bacterium that is able to divide and form these aggregates and also make some form of metabolite that allows it to be more metabolically active in the anaerobic part. And so this is wild conjecture. This is a hypothesis that I've had, but I'm hoping that with the tools I'm developing, I can figure out what bacterium is there. So all that is to say, so far I've only showed you fish with generic, so like a universal probe for bacteria. The idea that I've been working on is basically how can we figure out specific genus or family probes that can tell us the different types of bacteria that are in the chicken gut. And so here, after many months of optimizing and designing probes and figuring out whether the probes work, I'm showing you here is again a healthy chicken 1 with its ceca with the most diverse bacteria. And I've been able to decode basically these different bacteria that are present in the healthy chicken ceca. And this is in tandem with our 16S data because basically what you find to be the most abundant in 16S or in metagenomics, you would expect it also to be the most abundant in our sections. But one thing that 16S doesn't do as well with fish, and that's why we chose to do fish, is that you're able to see not only morphological, but also localization of bacteria next to each other. Because if you just do 16S, you'll never know whether A bacteria and B bacteria are ever next to each other. But in this study, I was also really excited. When you look at this slide here, the green bacterium, F. proutzi, in different parts of the cryosection, it actually takes on an entirely different morphology. And so here I'm showing the same sample, same experiment, just an image from a different part of the cryosection. You now see that this green bacterium becomes filamentous. And at first I was a little bit like, okay, my probes are wrong. This is an entirely different bacterium. I did something wrong. But through literature search, it shows that when F. prout is grown to stationary phase, it does form this filamentous lifestyle. And so I was really excited because it meant that fish is working, number one, but also that you can detect morphological changes that you normally cannot do with just metagenomics. And also, I told you I was excited about these aggregates. We also found bfrag, this blue bacterium, forms aggregates in the chicken gut. And so this is an example of why fish can tell you a lot more information, a lot more 3D information that other types of methods don't usually show you. And the last thing, I also carried out fish on a salmonella-infected chicken. And mind you, I've been using the same exact probe combinations for both samples. And what you see is here this orange bacteria, Lactobacillus crispatus, that is found in the healthy chicken is now entirely depleted in the salmonella chicken. And so I was really excited about this finding because if you link it back to our 16S analysis, we showed that Lactobacillus gets depleted in the salmonella chicken, and this comes hand in hand in our fish analysis. And so all of this was really exciting to me, and I really can't wait to see where this type of question and this project will go as we improve our resolution and all of the tools that we're working with. And that's mostly what I have for you guys. And I'm bringing back this photo again that Antton had showed because I cannot emphasize enough how exciting it is to work in this project, because both what Victoria and Jenny had talked about, I have no skills in that area, but I get to participate in this project because we're bringing together truly experts in their various fields to work together. Yeah, and so with that, I would like to thank everyone for your attention. And I guess it's questions time.
Thank you so much, Bryan. And thank you for the lovely enthusiasm. It's really inspiring. I love those pictures. They look really cool. It's all very scientific so far, so I'm really interested to hear. Maybe our audience has questions for our presenters today. You're open to post them in the chat or open your mic. But while we wait for that, as I mentioned, it's quite scientific. So what do you expect will be the practical implications of the results that were presented today, or the plans that are presented today? Do you have an idea about that? Because I think the TRL level of solutions or ideas that 3D-omics might propose is still at the beginning stage, or how does that work?
Shall I respond to that? I would say we have, let's say, we are advancing in two phases. So we are, on the one hand, generating and developing a new technology to reveal interactions that have been hidden so far. So not even, we didn't even consider would be possible to get to this level a few years ago. But at the same time, we are also generating multi-omic data that it is far closer to the kind of information that we are now getting more and more used to for improving, let's say, farming practices or breeding practices, and so on and so on. So our hope is that we will develop the multi-omic, the microscale multi-omic technology almost to the level in which the macro-scale multi-omic level so technology is currently, and hopefully that can give us new ways to find hidden links between microbes, among microbes, between microbes and hosts, etc., etc. It is true that for the, let's say, for companies that need to implement a new technology, also a new strategy right now, we're still far away from that point. This is something that probably will yield practical implementation in 5-10 years from now. That's more practical, because we are not sure either how deep we can go into resolution and how scalable this is. So these are the kind of things that we are assessing. This is the last part of the project is about. It's about assessing what is the extra information we can get, for how much extra time and financial investment we can get that new resolution. And hopefully we will get all these answers by the time that the project is over. And it's going to be the aim of a following project to take this to the very practical landscape of helping farmers and additive producers, etc., to implement this knowledge in their everyday practice. Thanks.
Yeah, because of course 3D-omics is the sister project of HoloRuminant, and HoloRuminant is doing, it's more a frontier work of figuring out what we have, what we can do, and what we can get to. So I'm hoping that indeed we'll both get the answers we need, and that we can get the follow-up that will be beneficial for all of us. Are there any other questions maybe from the audience? If there are none, then I'll try to find my presentation again. This is recorded and I will also share it online. So if there's any questions after the webinar, you can also post them maybe under the recording when it's posted online. And I think we can keep an eye out to get the questions to the right people. Let's see if the sharing works. This one. So thanks again for all speakers. It's really enlightening, and I have seen some very interesting and new things that I haven't learned about before. So that's really cool. With that, I would like to point out that there's going to be a third webinar in the Joint Dissemination Network series. It will be on the 3rd of October. And we'll have presentations from the Geronimo project and the Ruminant project. And specifically from the Geronimo project, we haven't heard anything in the series so far. And the theme of the upcoming webinar is basically social acceptance and stakeholder perception. So it will be a little bit less technical than today. And with that, I would like to thank you for participating today, and I hope to see you all on the 3rd. We also have an international stakeholder platform. You can scan the QR code here to join it, and then you'll stay up to date also when the recording becomes available and so on. So with that, I don't want to keep you any longer, and I thank you for your participation today, and I hope to see you next time.